So thanks for the organizer to give me this opportunity to present my work. So in the last 2 days, I think we've seen really fantastic talks. They all revolved around single-cell sequencing technology. And now in the next 25 minutes, I'd like to talk about another technology which lets us see a whole living organism in its entirety with imaging at the cellular resolution. So we're now switching to microscopy essentially. And so the kind of microscopy I want to talk to you about today is Latchet microscopy, which is it's a relatively novel form of microscopy that has the advantage of being gentle, fast, and volumetric. So what we do in Latchet microscopy is we create light sheets that basically illuminate a fluorescently labeled sample. And by doing so, by looking at exactly what we illuminate and only illuminate what we look at, we can achieve a very gentle imaging over a very long period of time. So we can image living embryos, living larvae for hours and days. And this is a very powerful method to basically study living systems. And so when you do that, of course, the first step is to acquire the data. But that's not nearly the last step. You have many other steps that you have to perform. You also have to ideally visualize your data in real time as you acquire the data so that you can actually decide whether that's actually a sample that is worth imaging or not. If you visualize, you also have the opportunity to analyze this data in real time. If you can do both, then you have also, this affords you a unique possibility to adapt your microscope to the differing optical conditions of the sample across space and time. And so you can basically potentially have a microscope that's self-driving and can really push the resolution to the limits offered by optics. Going forward, you can imagine also leveraging the recent advances in artificial intelligence that let you improve the quality of images, potentially. And so I will talk also a bit about this, how you can actually use the latest results in deep learning to push further the resolution of these images that we obtain with the microscopes. So this work is essentially, so what I described is essentially what we strive to achieve with my colleagues and collaborators. And in the next 15 minutes or so, I'll try to discuss more the later two projects, which are essentially Autopilot and KARES. The first one is how to turn these sophisticated light sheet microscopes into self-driving systems that can push the resolution and maintain resolution throughout a long time lapse. And the second is how to apply deep learning to imaging. So first, the Autopilot project. This is basically the kind of data you can acquire with these light sheet microscopes. This is really a three-dimensional data set. And so you can, for 21 hours, image a whole living Drosophila embryo and follow almost every cell throughout this time lapse. And we do that every 30 seconds. Every 30 seconds, we have a new image, a full 3D image. And so this is a lot of data, a terabyte per data set. And it opens up a lot of interesting questions in terms of how does such an organism start from a few cells all the way up to a very complex system that is then twitching and then eventually comes out of the microscope alive. And you can see here that the resolution is quite high. And the question is, how do we achieve this image quality in the face of so many things that can go wrong? So the question is, what are the things that can go wrong? Well, the way we mount this sample is we put the embryos or larvae, depending on the specimen that we're looking at, we put them inside of an agarose gel. And because of the different refractive indices in the water, in the support matrix, which is typically agarose, or the specimen itself, you have the light that enters the specimen and leaves the specimen is bent and distorted. And so this causes basically loss of image quality. And so you have to take that into account and somehow correct for it. Similarly, within the specimen itself, there are variations of the refractive indices over space and time that conspire to degrade your image quality further. And if you actually do a multicolor imaging experiment, you have to do these corrections potentially for different wavelengths. And sometimes you cannot do these corrections in advance because there's just simply no signal. For example, here, if you have a paneural imaging, you see that the neural system is labeled here in red in this figure. And so the neural system appears halfway through the time lapse, and so you cannot adjust the microscope in advance. All right. So the same thing happens if you have migrations of cells throughout an embryo. Here, a zebrafish embryo. You have essentially what the process of epibole going on here over 6 hours or so, and as I mentioned before, the same problem occurs here. You cannot adjust your microscope to these regions here that are void of cells, and so you have to have a system that's capable of detecting migrations, detecting the onset of expression, and adjust itself automatically. So the microscope at the schematic level looks like this. You have multiple detection and illumination objectives, and really the challenge is to make sure that all the degrees of freedom, so all the knobs that we can turn to adjust the microscope, that they're all under first digital control, so the computer can actually tweak these parameters, but we also have to have ways to decide what these parameters should be. And so, long story short, there's a whole theory that we developed to actually formalize that and solve the problem. Every 4 minutes, we have a microscope that can basically readjust all these parameters to guarantee the best possible image quality over 20 hours of imaging or even longer. And so let's have a look at the microscope in action. So here you have basically the developing embryo, the two sides, the dorsal and ventral sides, and you have basically parameter time courses that show you how the parameters of the microscope have to be adjusted at different imaging depths and over time to maintain the best possible image quality. And so you see here that there's a lot of variability, and so there's really a challenge to be addressed. The question is how much better it is if you apply this, whether if you apply it or not. So here, if you have the correction on, you get this image quality here. And if you do not correct, then you have this quality. And so you see quickly, over time, it degrades to the point where in some regions, you can see the cells individually. And so you wouldn't be able to follow them over time. You wouldn't be able to do a fate mapping or linear tracing over certain regions. The same thing in zebrafish, where it's even worse because, of course, we have not even had the possibility to adjust the microscope in advance for certain regions. And so here you see that the quality is dramatically reduced over time, and these curves here tell you, like, basically show you how much the degradation is getting, how much it's how it gets worse over time. And so, luckily the system is capable of correcting all that over time, and so we basically overall we get basically this image quality here. So this is basically, we can maintain the quality of the images as optimally over time. In the case of a multicolor imaging session, you can also do the same thing. In this case, the microscope is detecting the onset of expression, and here over several different planes of imaging, it detects the onset of expression and adjusts itself so that the processes of the neurons here are at the highest possible resolution allowed by the optics. And so we can do this now. We can build a microscope and the software into an integrated system that guarantees the best possible image quality over time. But the question is, can we push it further? And so this is this KARE project that we started with my colleagues in Dresden, and it's been quite a success. And at the core idea of this is really that where we started is that there's in microscopy this key problem that we would like ideally to maximize all these things at the same time. We would like to have the best signal-to-noise ratio, the best spatial resolution, the best temporal resolution, the best, to have we would like to image in the most gentle manner so that we don't kill the sample or we don't bleach the fluorophores. The problem is we cannot do all these things at the same time. We have to always have trade-offs. We have to give up something to gain something else. And the question is, can we solve this and somehow get everything at the same time, or at least not have always to trade off one for the other? And so here's an example. So if you try to image a live planarian, and you want to have actually good image quality and good contrast, the result is that you get indeed high contrasted images, but the planarian, they don't like light. So they will twitch incessantly if you try to image them this way. And so the result is you have a lot of motion blur. So you have high contrast but motion blur, and so the images are nearly useless. If you reduce the amount of laser power and also reduce the exposure, you reduce the total amount of light by a factor of 60, and now you have much less motion blur, so much that each frame is pretty sharp in terms of like motion, but the contrast is very, very poor. There's a lot of noise, and so now the images are useless, but for a different reason. And so how can we resolve that problem? So we as many others, and I think we saw some presentations yesterday about deep learning also, we thought of using these recent advances in deep learning and machine learning to basically try to find a solution to this problem. And if you've seen in the news the amazing results of DeepMind in basically addressing for the first time beating a human at the ancient game of Go, you might have noticed that one of the key technologies behind this was, among others, the usage of deep convolutional networks. And this core idea is very interesting because for the first time, the key thing here is that in contrast with Deep Blue in 1997, when IBM managed to beat Kasparov, the solution that DeepMind took is something that's more akin to artificial intuition. These networks somehow have the same capability as our brain, if you want, to basically capture something very profound about images. And so we use these convolutional neural networks to try to basically convert, recover the signal from the images. And so the starting point of that is you have to have example images. You have to have pairs of images, the ones that you want and the ones that you have. So this is what we acquire, very low signal, but we know the information is there, and we want somehow on artificial neural networks to basically give us back what we would like to actually use. And so there are many ways you can actually obtain these pairs of images. That's a key problem. How do you get these pairs of images? So one solution is simply to fix the sample, acquire pairs of images at low and high laser power, and now because now the sample is not moving because it's dead, you can do it. And so now you can acquire this training data on a sample that you will not use for science, but you'll use it for learning how to better see. And you can also use other elegant strategies, like for example, you can use simulations to get these pairs of images. And so this is really the core questions. How do you get these images? And so in our paper, we have shown a few strategies and applied it to a few problems, like the problem of denoising these images. And the result is pretty stunning, that you can train these networks to basically go from images that look like this into something that looks like this. So you're almost at the point where you beat the human eye at seeing, and the human retina and a few layers more at actually recognizing patterns in these images. If you really take some time, and we have done that, you can see why this is explained by this. But it's very subtle, right? And so if you do that, now if you train on these fixed samples and you take these pairs of images and feed them to the network and tell the network, please learn to do that, then it will actually manage to turn an image that's highly noisy and where it's very difficult to distinguish the nuclei into something that is much sharper and very clean. And so when you apply that over an entire 3D data set, this is what you get. And I'm very sorry, there's some technical problem here with the connection. The resolution you see on the screen is quite reduced. We can barely see the last name of Martin Weigert, who was the first author on this work. So what happens here is like with a total reduction in light dose of 60-fold, we can still get the same image quality. And so we apply this little trick. So the great thing is that with deep learning and machine learning in general, now we write an algorithm once, and problems are then defined by the training data we feed to the algorithm. So we can solve other problems, like for example, improving the resolution along the axial direction of microscopy images. So in microscopy, even in electron microscopy, there's this big problem. How do you, there's this big problem that you cannot get nice as nice a resolution along the z-axis as along the xy planes that you acquire. So there's this anisotropy inherent to microscopy that we try to address here. And by training, by learning from the xy slices and applying that along the x, z, and y, z directions, we could basically recover the resolution so that now we have a 3D stack that is completely isotropic. And of course, I mean, it relies on the fact that these images have a well-defined structure. The same trick for super resolution, where you have now wide field, and then we basically manage to recover something that is akin to super resolution. And we use simulations of microtubules and granules and simulate all the optics and use that to basically do the inversion and somehow deconvolve these images in a far smarter way than we could do otherwise. So we use like the knowledge that we know what is there, but we just want to see it better and see the dynamics over time better. So what is next? So started 1 year ago at the BioHub, my lab. Chinmay has joined the team. Ahmed and Merlin are joining soon. We already have a microscope that's just requires a few finishing touches to be ready. And so I hope to soon be able to give you an update on these discovery machines that we're trying to build. And I'd like to also acknowledge all the people, and I don't think, you know, there's quite some more people that I could put on these slides. So all this work is of course like teamwork. But I'd like to basically mention 6 people in particular: Philip Keller, Raghav Chetri, Bill, Jean Myers, Martin Weigert, and Florian Yuk, who have been like very forefront on all this work. And of course all the people at BioHub and for being fantastic colleagues over this year, and you for your attention. Thank you.
So you're basically using deep learning to manipulate the images.
Oh, here. Sorry.
In light of recent papers describing the lack of robustness of those deep networks, like, for example, the recent paper that shows that just a single pixel attack can actually change the classifier results, aren't you worried that you might generate artifacts?
Yeah, so, you know, this is something that is always of concern, and I think like with very, very powerful tools, you have to be even more careful. So I think there's a lot of work that needs to be done in terms of like quantifying the uncertainty of these networks. And so this is opening a new kind of whole direction in research here. How do you basically have networks that don't just produce better images or do a task better than was possible before, but how do you actually make sure that these networks also tell you something about how accurate is the prediction? And so yeah, it's the problem is the explanations are probably not understandable by humans. So, you know, at some point, I think every microscope, every technique that observes will make some mistakes in one kind or another. And I think the most important thing is in terms of education, and making sure people understand the tools they use and understand their limits, and understand also how to use them and how not to use them. For example, you do not train a network on images of nuclei and apply that on microtubules. Actually, the networks are pretty smart. What we observe is if you do that, actually refuse to predict anything. But in some cases, they might get confused, and so you have to know how to use these tools properly. And it's also a challenge, I think.
Hi, Loïc. Nice presentation. Of course, I think it's very clear that deep learning-based algorithms will replace all filter-based, which has been used so far, because they introduce much more bias. Clear that. So related to your algorithms that improves the Z resolution is isotropicity. That's, I think, very cool thing for light sheet microscopy especially. So two related question is how applicable this to different the images coming from different zooms or different structures, like if it's neurons and vessels, how much additional training would be needed to adapt these algorithms? And do you have plans to somehow, make these algorithms that everyone can somehow improve it by their training data, like collectively to train it better and better, that it's solution for all light sheet users?
Yeah. So for the isotropic recovery, the interesting thing is that we do not train on anything else than the stack that you start from. So it's a little bit of a non-common situation where usually you train on this huge data set and then you apply it on some data. This is not what we do. We have one stack. Actually, any stack you ever acquired, if you know the optics and the sampling characteristics, you can apply this algorithm to it. We learn on the same data set that we apply it to. We learn on a different aspect. We learn on the fact that we have planes of high resolution, but we have too few of these planes, and then we just apply it, if you want, by rotating the situation. So that means that any stack that has data that is inherently isotropic, and a few more conditions that you have to be aware of, then you can apply it. You can apply it to this data. Yeah.
Hi, that was a great talk. I think this is great, especially for engineering type applications. But I've also been a bit wondering about scientific applications where you're trying to discover something novel. What if that thing you're trying to discover is not represented in your training data set? How do you figure that? And then also, how do you educate users as to what they can and can't conclude?
This is a very nice question, and actually this is a question that I get every time. So I have a very well-prepared answer to it. So here's the way I answer this. One simple analogy is that of a book that would be written in an alphabet that you know very well. But the book is so old and so weathered that you cannot see the text well. So what deep learning can do is it can take that book where you can barely see the letters and the words, and it can make that much easier to see. And so, of course, the alphabet is not what you're actually trying to discover. What you are trying to discover is what the book is saying. But if you know the alphabet, you can train a neural network to make that text more visible, and then you can discover what the text is saying. And in the case of microtubules, for example, in the last movie, the movement of the microtubules, the temporal dynamics is maybe what you're after. You're not after discovering a new kind of microtubule, but you want to see them better. And so even if you actually use existing knowledge for analyzing your data, it does not mean that it precludes you from discovering something new. If discovery would require not knowing anything, we'd probably not discover anything in the first place.
All right. With that, let's thank Loïc again.
Thank you.